This recording is going to talk about transport across cell membranes. There's already a recording about phospholipid structure, and so that tells you how and which molecules will get across and why. And this was just going to talk about the different types of transport. So you can see here that there are different ways molecules get across the membrane. So some molecules are going, or I should say solutes, because not everything crossing the membrane is a molecule. Some of it's ions. Some solutes can cross the membrane directly. So you see these are going straight through the phospholipid bilayer. They don't have any helpers getting them across. This process is called simple diffusion. Diffusion is the tendency of particles to spread out into empty space. Being spread out is a very stable state. It's a low energy state, and biological systems move towards low energy states unless there's some other type of energy put into the system. So the natural tendency of molecules and ions, all particles, is to diffuse and spread out. So simple diffusion is one way of spreading out. We have scenarios where the solutes can't get through on their own. So they need helpers. So there's two different types of helpers here. They're both proteins. There's one that's a channel. We call that a channel protein. And then there's one that changes shape. We call that a carrier protein. And both of those, this also is diffusion because it's going and spreading out the molecules from the concentrated to the less concentrated. But this time we have a helper, so instead of simple diffusion, we call it facilitated diffusion, meaning diffusion that's aided by the proteins. Both of these processes are spreading from high concentrations to low concentrations. And really we're just looking at, so it's high purple to low, high triangles to low, high blue to low. So each one spreads out with its own type. And all this spreading out from high concentration to low concentration is what we call passive transport. So passive transport is not going to require additional energy, and is basically using the energy of the concentration gradient. There's already energy in the system because you have solute on one side but not the other. So it's sort of the idea of having water behind a dam. So lots of water on this side, not on the other when it flows through. So there's lots of potential energy in the system, and so that energy is what's used to move it. So it's not any additional energy into the system. That's why it's passive. Then we have transport type 2, which is when we go against the concentration gradient. So in this case, we're moving the diamonds from where there's more concentrated diamonds to less concentrated diamonds. And the same with the circles. The circles or the spheres are going from there's also a process called active transport. It's the another type of transport. So in this case, we're going against the concentration gradient. So in this case, you can see here's our diamonds. The diamonds are going from where they are less concentrated to where they are more concentrated. So that's going to be like shoving more solute into the side that's already concentrated with it. So same thing with the spheres here. The spheres are going from where they're less concentrated, and they'll get pumped over to where they're more concentrated. So this type of transport against the concentration gradient is called active transport. And that's because it's a process that requires additional energy of some sort. This is representing that. Frequently it's ATP, but not always. And you can understand that because pumping against the gradient is basically like you're trying to put more water behind the dam. And in order to do that, you have to some sort of pump. It's the same scenario here. So we always have a transport protein. We can call it a pump. And it's going to be pumping solutes into where they're already more concentrated. So just a brief overview of what can pass across. So molecules like this that are small and nonpolar, lots of gases, or all gases really, any kind of hydrocarbons, for example, steroids, monoglycerides, fatty acid chains. Remember, steroid examples are cholesterol, testosterone, estradiol, that kind of stuff. All those are going to be able to pass across. And so this would all be by the simple diffusion, passing directly across because you are nonpolar. What can't pass through? So molecules that are polar, so partial charges, charged particles, those would be the ions, and other hydrophilic substances, like glucose, which has some polar parts to it, the hydroxyl groups there. So these will pass by facilitated diffusion. Now both of these are assuming that we are going from high concentration to a low concentration, so simple diffusion and facilitated. So both of these scenarios are passive transport. You can't get too big. If molecules are too big, like a big protein or a starch, they can't get across the membrane themselves at all. So they're going to have to do bulk transport, which is called endocytosis when it's going in, or exocytosis when it's going out. So here's our little chart. We've kind of talked about it. So passive transport goes from high concentration to low, the natural tendency, versus active, which goes from low to high, shoving into an area that's already concentrated. That means, I think of it as a triangle. If we're going from high concentration to low, we're going down the concentration gradient. And if we're going from low concentration to high, we're going against the concentration gradient. The concentration gradient just means the difference in concentration across a membrane. Energy requirement. No additional energy required for passive. Additional energy required for active, frequently in the form of ATP, but not always. Then we come to transport proteins. So passive transport, the two types, simple diffusion does not use transport proteins, but facilitated diffusion does. So we could say sometimes passive transport uses them. To pump against the gradient, you'll always need a transport protein for active transport. What types of proteins? So channel proteins and carrier proteins can be involved in passive transport, facilitated diffusion. In active transport, it's only carriers, which we sometimes call pumps. It can't be channels, because channels are just corridors through which solutes pass, so this would be the channel, and so that can't go against the gradient because it's just flowing. Subcategories, we said simple, so that would be things like the gases. We said facilitated. That would be, some examples of that are water movement, so aquaporins. We have glucose transport. And those are some amino acid transport from high to low. And then examples of active, a lot of times we talk about the Sodium-Potassium Pump. It's important for neurons. We talk about hydrogen ion or proton pumps. Those are going to be important for photosynthesis and cell respiration. Just to remind you that even though the molecules spread out into space, diffuse, they individually move randomly. So sometimes they bounce off the membrane. Even after they have moved to one side, sometimes they'll move back. It's all random movement, so it's called Brownian motion. So it's the random vibrations and the knocking of the particles into each other that causes them to spread out eventually, but the molecules move randomly themselves. And orange only diffuses where orange is lower concentrated. Purple will diffuse where purple is less concentrated. So they don't depend on each other for diffusion. A special type of passive transport is called osmosis. So osmosis is when we're talking about water as the transported molecule. So this is instead of a solute, which is the substance dissolved in water, we're talking about water itself. And so you can see the water molecules here, and here's some solute. And so so solute is more concentrated on the right side than the left, which means water will go towards where there are more particles per volume, so higher concentration. Water moves towards that side to dilute the concentration. And we use terms to describe the conditions of the two sides compared to each other. We call those tonicity descriptions, or the tonicity. And so there's three main terms to use with tonicity. So one of those terms is hypertonic. Hyper means lots, so like hyperactive children have lots of energy, too much energy. So hypertonic means you have a higher concentration of solute compared to another solution. So you'll never just be intrinsically hypertonic. You could have an 80% glucose solution, so that's very high. But if you're comparing it to a 90% solution, then it's not hypertonic. So it depends on what you're comparing it to. So hypertonic, we draw a box and a membrane inside the box. If I drew it like this, with this solute, we would say that the inside of the membrane is hypertonic to the outside of the membrane. We could have drawn it the opposite way. Here, the outside of the membrane is hypertonic to the inside solution. Hypotonic is the opposite of that. So hypo means below or underneath, like hypothermia, when you have too little body heat. Hypoglycemia, when you have low blood sugar. So hypo means low. So the hypotonic solutions are going to be the ones that are less solute. So in this case, if we were comparing the environments is hypotonic to the inside of the membrane. And in this case, the inside of the membrane is hypotonic to the outside. So it just depends on what you're describing. So I always suggest you draw pictures. I'll tell you something is hypertonic or hypotonic, or I can tell you, you have 20% glucose on the outside, you have 10% on the inside. So then you draw that up, just drawing more dots on the outside because it's 20%. And then you can think of where water's going to move. The last term is isotonic. Iso meaning equal, like an isosceles triangle with equal length sides. So isotonic means that both solutions are going to have equal concentrations of solute. They'll be in dynamic equilibrium, moving across the membrane in both directions at the same rate. So if we look at the scenario here, we have two solutions separated by a selectively permeable or semi-permeable membrane. If we describe the solutions compared to one another, we look at the one on the left side compared to the right. The one on the left has less particles per volume than the one on the right. That means the left side has a lower concentration of solute than the right side. So what we would say would be the left side would be hypotonic to the right side solution, which leaves us with the right side solution being hypertonic to the left. So you see we have to compare them to each other because neither is intrinsically hyper or hypotonic. Now if the solute, the purple circles, could just even themselves out, they'd move from the right to the left and that would even them out. But let's say in this case the selectively permeable membrane doesn't let the solutes through because they're too big in this case. And this could also be in a cell where they're charged or something like that. They can't get across the phospholipid bilayer. So the membrane is not permeable to the solute. We could also call the solute non-penetrating. It's a non-penetrating solute. So what happens? Instead, so water's going to move instead, and so what water does is water moves towards the right in this case, so it's going to move to dilute the more concentration, the higher concentration of solute. So this moves to the right, that means that your water level goes up on the right and down on the left, so you diluted the right and more concentrated the left, and now they should be equally concentrated. So now, if it's at equilibrium, they'd have isotonic conditions to one another, where water would be going back and forth across the membrane at an equal rate in both directions. So that's what osmosis would be, the movement of these little waters through the semi-permeable membrane. So for the exam, you need to know how to work YouTube problems like this. You'll also want to practice some dialysis tubing problems. There are ones on D2L. You can look up tonicity problems online for practice. There's Mastering Biology. There's definitely ones in the book. If you have a study guide, those are of them as well. So there'll be kind of scenarios where talk about hypertonic, hypotonic, or different concentrations of solute. So let's say 20% on the outside and 10% on the inside, and it's non-penetrating, and you have to say what's going to happen. So in this case, the water would move in, and depending on the cell type, the cell would behave in certain ways. So what happens to different types of cells and they're placed in different environments? So the animal cell is just a generic cell without a cell wall. So any cell, whether it's animal or not, that does not have a cell wall will behave in this fashion. Plant cells are representative of a cell with a cell wall. So any organisms or any cells with cell walls will behave in this fashion. So if I tell you something and give you a hint that this organism does not have a cell wall, or I expect you to know that fungi have chitin cell walls, then you could decide whether it would act without a cell wall or with a cell wall. So if we place an animal cell without a cell wall in isotonic conditions, the water is going to go out and in at the same rate. So it's going to stay at the same volume, and so that will be very healthy for your animal cell in isotonic conditions. In hypertonic conditions, what we've done is we have a lot more solute outside the cell than inside the cell. That's because it's hypertonic. So what does that mean? Water moves towards where there's more solute, which means that water is going to move out of the cell into the environment to dilute the environment. And this can go on for a little bit and we shrink until it reaches equilibrium, or if it goes on even longer than that, the cell will actually shrivel up and die. Then if you put an animal cell or a cell without cell walls in a hypotonic environment, that's going to result in the cell taking on water. So in this case, there's more stuff inside, more solute inside the cell, less outside in the hypotonic environment. So that means water is going to travel in to dilute the concentration of solute inside. First, it will swell, cell swells, and perhaps that's all that happens if it reaches equilibrium. But if it doesn't and it keeps on swelling, eventually it'll lyse, which means to burst. So that's what's happening here. In plant cells, it's a little different. So plant cells in isotonic conditions, water going in and out at the same rate. You can see the central vacuole holds most of the water of the cell. It's actually not a good condition for plants. They are flaccid, which means limp or wilted. And so this is because plants really need a lot of pressure on their cells to keep them rigid and keep the plant upright. So the condition where we have rigidity is really our hypotonic environment. So there's more stuff inside of this cell than there is on the outside, because the environment is hypotonic, which means that the water is going to move into the cell and into the central vacuole. That means the central vacuole swells, it puts pressure against the cell walls, which is going to keep the cell very rigid and keep it firm. It's called turgid. The cell is turgid, and that means that the plant is going to be able to keep itself upright. If we put a plant cell with a cell wall in a hypertonic environment, we have lots of solute on the outside, much less than the inside. Lots of solute on the outside, much more than the inside. That means the water is going to move out of the vacuole and out of the cell in order to dilute the environment, which means the cell will first shrink a little bit and then it undergoes plasmolysis, or it plasmolyzes, which means that the cell wall actually pulls away from the cell, which means the cell membrane actually pulls away from the cell wall, and the cytoplasm shrinks up and the cell will the cytoplasm shrinks up and the cell will die. So this case where it's hypertonic, that's like when you really have a problem with your plants. Isotonic, they're wilted, but you can bring them back from that. Hypotonic, they're the healthiest there. Here's an example of a dialysis problem you might have. In this case, I would tell you that the membrane is selectively permeable and that non-penetrating solutes sucrose, so it's permeable to glucose and fructose, so the monosaccharides in this case. So if we look at, we'll look first at glucose. So glucose can get through the membrane, which means that it'll move itself to even out. So if we look 0.02 molar glucose, that's just a measurement of concentration, 0.02 inside and 0.01 outside. So that means that there's more glucose inside than outside. So the glucose will move out into the beaker until the concentration's equal. So until each concentration is 0.015 molar glucose inside and 0.015 molar glucose outside. What happens to the fructose? So fructose is going to be able to move itself as well. So it is also one of these that can penetrate the membrane. So fructose is 0.01 on the outside, nothing on the inside. So that means the fructose will move from the environment into the inside of the cell, and until there's 0.005 outside and 0.005 molar inside. So it'll even itself out. Then we get to sucrose. Sucrose is a non-penetrating solute. It can't get through. So what's going to happen with sucrose? It stays where it is. However, it's still not in equilibrium. Water will move into the cell in order to dilute the 0.03 molar sucrose until it gets to 0.02 molar, which will be equivalent to the amount of 0.02 molar on the outside. And so now both of the sucroses are equal as well. So when water moves in, that means your cell is going to increase in size. It'll swell a little bit. If it reaches equilibrium without having to add too much water, then it will just swell. If not, it could actually burst if it takes too much water to try to make it to equilibrium. Let me just point out this is facilitated diffusion now, of which water undergoes facilitated diffusion. Water primarily travels through a pore called aquaporin. So that's a type of channel. The two types we talked about a couple slides ago, so channel proteins and carriers. I just want to point out that there are multiple kinds and they're very specific. So aquaporins only let in water. Different ion channels only let in certain ions. So like a sodium channel, Na+, will not let in a potassium ion, even though it's still just one atom and it's charged, plus. So they don't let each other in. They're very specific. And finally, what about solutes that are moving against their concentration gradient from low to high? That's going to be active transport. We've already seen the basics of that. So the sodium-potassium pump is a good example of active transport. So you can see it involves a carrier protein, as always. In this case, we have sodium that's being transported from where it's low to where it's high outside the cell, and potassium being transported from where it is low to where it's high into the cell. And so those are both against their concentration gradients. Pump is important for building up concentration gradients so that your nerves are able to signal. So this is how a nerve works. Here's the sodium-potassium pump. Lots and lots of sodiums outside, lots of potassiums getting pumped inside. Then you have channels that open at different times in nerve firing, and when the channels open, some of them are potassium channels, so potassium flows back outside the cell. Some of them are sodium channels, so sodium flows into the cell. And this basically changes the charges across the membrane because it moves the ions, and that makes sure the next one open up, and the next one open up, and the next one open up, and it basically moves your signal down the nerve. Pumps that move ions are called electrogenic. So basically it's because they create a membrane potential, which is when you have positives and negatives in different amounts on the sides of the membranes. So the chemical force is the ion itself's concentration gradient. So where is sodium high, where is sodium low. Electrical force is the plus minus, and where's the pluses and minuses distributed across the membrane. Pumps in general are quite useful for creating concentration gradients across the membrane. So they'll pump in this case the ions to one side in large amount over the other, and so you'll have unequal amounts of ions, and so basically it's like water behind a dam, that kind of analogy, where the water is poised to come out of the dam to do work. And so the same thing here, you poise the ions in one side or the other of the membrane. You can also do this with other molecules, but active transport's what's used to get them there and make this steep concentration gradient, so it can do work later in the future. So why does that matter? So here's a sodium-potassium pump, and here is a sodium channel and a potassium channel. This is how nerve signaling is going to work. So the pump is going to pump the sodium out to where it's already high and potassium in to where it's already high. You'll notice that you put 3 sodiums out for every 2 potassiums in, which means overall you're getting 3 pluses outside and 2 pluses inside, which overall makes the outside of the cell positively charged and the inside of the membrane of the cell negatively charged. So how's that going to influence sodium and potassium when they try to move back across the membrane? So let's look at this. Diffusion of sodium ions here is facilitated by or impeded by the sodium concentration gradient. So we have to look. Okay, so sodium is going to be moving this way. Is it promoted or impeded? So we look going from high to low. So that would be that it is facilitated or helped by the concentration gradient. Now let's look at the potassium. Potassium moves in this direction. So is that facilitated or impeded by the potassium concentration gradient? It's going high to low potassium, so that one is also facilitated. So the chemical force in this scenario helps move both the sodium and potassium ions passively across the membranes. So what about when we look at the electrical forces at work here? All right, so let's look at our sodium ions again, and we're going to look at charges. So the sodium ions are positive. They want to come inside. So inside's negative. We know that opposites attract, and so that's going to again be facilitated. So what about potassium? Potassium is positive. Potassium wants to go out. And so look at this though. Potassium's positive going towards the positive. So that means that potassium is impeded by the electrical gradient. So overall, both of them have a facilitating chemical gradient in their respective directions. But sodium also has a facilitative electrical gradient, whereas potassium has an impeding one. So overall, the sodium has an electrochemical gradient that's larger than the potassium because both the chemical and the electrical add together. So that just means that sodium is going to flow into the cell more easily than potassium flows out of the cell. And these are both passive transport channels. The active transport just continues to pump in the direction that keeps building these electrochemical gradients. Proton pumps are also electrogenic because they are pumping ions, so the positive charge is going to build up in this case on the outside in the extracellular fluid, and in this case the negative is on the inside. This is an active transport, so it does take energy, in this case in the form of ATP. So this is called cotransport. So cotransport is a form of active transport, and that is because it uses energy and moves solutes against their concentration gradients. So here is energy usage, and it's pumping hydrogen ions outside the cell, protons. Those protons then diffuse back into the cell. That part's passive transport. But as they diffuse into the cell, in this case, they can take sucrose along with them. Sucrose is going from low sucrose to high sucrose, making this part active transport, low to high. So we have active transport because of the ATP for the pump. We have active transport because we're putting the molecule that comes along for the ride for the cotransport. That's going from low to high. So it doesn't have to go both in the same direction either. Some cotransporters, one goes in and the other one comes out. Both of those are still the same. Basically, it's relying on a concentration gradient, and as that flows back down its concentration gradient, it will do the active transport of the other substance. So here's an example of the sucrose hydrogen ion or sucrose proton cotransporter. So this would be one way you could get sugars into cells that are already full of sugar. Bulk transport is the last type of transport, and basically this is for moving large molecules across the membrane. or taking large quantities of small molecules in or out at one time. So this is when cells are going to secrete molecules or do things like ingesting molecules from the environment. Well, there's two different types, exocytosis and endocytosis. So exo, like exoskeleton in an arthropod, exo means outside. So exocytosis is dumping products outside the cell. So here's your cell, and here's your little vesicle filled with products. The vesicle is going to fuse with the plasma membrane, and then, like this, it will dump those products out of the cell. That's exocytosis. Endo means in or inward, so endocytosis is the opposite. So here's your cell. Here are some products that are going to get taken in. The cell makes a little pocket. We'll mark the membrane there that surrounds those particles, and then those are going to get pinched in into a little vesicle and taken into the cell. So that's endocytosis. So what types of cells do lots of endo and exocytosis? So exocytosis is going to be done by secreting cells, particularly those secreting proteins. So these vesicles are basically the ones that come from the Golgi, from the ER prior to that. Those are the ones frequently that are the ones that are undergoing exocytosis. So good examples of eating cells are amoebas and white blood cells. Both of them are going to intake particles in order to break them down inside. The amoeba will break it down for food purposes, and then the white blood cell will break down the virus or bacterium or other invader to protect the body. One type of endocytosis is called phagocytosis or phagocytosis. So this is the one that uses these pseudopodia in order to capture a particle from the environment, in this case food. This is like an amoeba surrounding its food. Then once it gets surrounding it, then the phospholipid bilayer rearranges itself and basically fuses with itself, and the inside part here will become its own little vesicle. And this is of course not open, this is a cut through. This is actually a sphere containing the food inside. So then what might happen when this comes inside with food? Well, that could fuse with the lysosome, and the lysosome hydrolytic enzymes might digest that food. Then the food can get shipped other places in order to be used either in cellular respiration or for building new cell parts. Pinocytosis is taking whatever solute is outside, taking it in the inside of the cell in relatively small amounts. So this would be, instead of having these extensions, the cell membrane just kind of folds inward and pinches off into these little vesicles that are going to take whatever's out there inside. Receptor-mediated endocytosis is similar to pinocytosis in the fact that it just pinches in, but in this case it has receptors that are specific for a particular substance, and so here are ones that are specific for triangles. So when you have receptor-mediated, you're going to get a lot more triangles coming in than in pinocytosis when you just get a random assortment based off of what's in the fluid. So receptor-mediated endocytosis is selective. So the LDL, low-density lipoprotein receptors, are on cells that help bind the cholesterol to take it out of the bloodstream and then ship it into, get processed into the liver. So that LDLs, those are going to be useful for removing cholesterol from the bloodstream. And different people have different amounts of LDLs, which can change how much cholesterol they can take out of the bloodstream and significantly impact people's health.